Hi class, welcome to the next lecture of the Medical Parasitology Lab. Today we're going to be talking about Plasmodium, or the parasite that causes malaria. So there's 4 species of Plasmodium that are considered true parasites of humans, meaning that they utilize humans almost exclusively as their natural intermediate host. They're Falciparum, Vivax, Ovale, and Malariae. P. falciparum is the most predominant species that infects humans, and it's the one that's most likely to progress to severe disease. There's another Plasmodium species known as P. burgei that causes malaria in rodents, and it's most commonly used in research, so it's very useful in finding new treatments for malaria for research. Mosquitoes of the genus Anopheles are the parasite's vector. So looking at the Plasmodium life cycle, it starts when a mosquito takes a blood meal from a human and injects sporozoites within the human. These sporozoites then infect liver cells, and then within the liver cells they mature into schizonts. When they replicate within the liver, it's referred to as the exoerythrocytic cycle. And then once the schizonts rupture, they then release merozoites. The merozoites then go on to infect the blood. So once the merozoites are released from the schizont, they then infect the blood cells. This is called the erythrocytic cycle, and within the erythrocytic cycle, they undergo asexual reproduction within the red blood cells. As they progress through the blood, they're referred to as the trophozoite, or the ring stage, when they're immature trophozoites. Eventually they mature further, and then they form a schizont in which they continue to reproduce. Eventually this schizont becomes so full of parasites that it eventually bursts the blood cell, and then the cycle repeats itself. And then what can happen alternatively is that the trophozoites can then differentiate into gametocytes. You have the female and the male form of gametocytes, the female being the macrogamete, the male being the microgamete. So these gametocytes will just circulate throughout the blood. Eventually another mosquito will come and ingest these gametocytes. They undergo exflagellation within the mosquito, meaning that they just mature more. And within the mosquito is when the gametocytes undergo fertilization, and then an ookinete is formed. Then the ookinete matures into an oocyst. The oocyst eventually ruptures and releases sporozoites. This form of sexual reproduction is known as a sporogonic cycle. And eventually the cycle then repeats itself when a mosquito then infects another human by ingesting sporozoites. So here is showing a more detailed image of the P. burgei sporozoites. So here you have the apical extreme with the apical complex organelles that are involved in secreting materials needed to infect the host. Here in this electron microscope image, you can see a more detailed image of sporozoites forming within an oocyst, and here labeled you can see all the apical complex organelles. So moving on to the schizont form. The schizont is seen in the blood when infection is severe. And then this is particularly a P. falciparum schizont, which can contain 8 to 24 merozoites. Here you can see in R is the rotrophies of the developing merozoites, the nuclei of the merozoites, and these asterisks just represent lipid bodies within the merozoite. So overall, something that's important to differentiate the different Plasmodium species is the schizont and how many merozoites are inside. So moving on to a comparison between the different schizonts of the different species of Plasmodium. So P. malariae usually has 6 to 12, can even be 8 to 10 merozoites. And there's P. vivax with 12 to 24 merozoites. So usually P. vivax has the most merozoites inside of it, and P. ovale merozoites are usually smaller, and they usually have fewer merozoites. They usually have the least merozoites of all the Plasmodium species. Moving on to a more detailed look at the merozoite. Here AP is the apical prominence. These other organelles here, such as the rotropy, are also needed to secrete the necessary proteins to help invade the host cells. You also see here the plastid, which is the apicoplast, which is something that is unique to apicomplexans, and also just helps in metabolism. So here in N you see the nucleus. Here in D would be the dense granules, also necessary for invasion of the host cells. The micronemes are also necessary in discharging contents to allow to aid in the host cell invasion. So moving on to the trophozoite stage, this is the stage that's found within the red blood cell of the host. So the ring form, which we see here in this image, is what's present in early trophozoite development. It's usually very thin, and that's how you can tell that it's a ring and not a more developed trophozoite. They also have a vacuole in the center of them, which also indicates that they're a ring form. So here this image is comparing younger trophozoites, which would be forming a ring, compared to older trophozoites of P. falciparum. And also note that P. falciparum trophozoites can deform the red blood cell. That's something that is important in comparing the infections between the different species of Plasmodium. And so something that we'll see in the next slide is something referred to as Schüffner's dots and Mahr's clefts, which are important in also telling the difference between the different species of Plasmodium. So here in Plasmodium falciparum we see the Mahr's clefts, and then here in Plasmodium vivax we see the Schüffner's dots. So the Mahr's clefts are just membranous structures that can be used for protein sorting and secretion of proteins that are involved in virulence, and the exact function of Schüffner's dots isn't really known. There's something that just is seen spread throughout the infected erythrocytes, but they may have some sort of metabolic function. So overall, Mahr's clefts and Schüffner's dots are just very important in differentiating the different Plasmodium species. So comparing the older trophozoites between the different species of Plasmodium, you can see, so P. malaria has two different forms it might be seen in. There's the band form and then there's the basket form. So in P. malariae, you won't see deformation of the red blood cell. So in the band form, P. malariae is kind of just spread horizontally throughout the red blood cell, where in the basket form it kind of takes over the entire red blood cell, and the chromatin here, just in the center, will be in this large mass. So the P. vivax trophozoite form will be amoeboid shape, meaning looking like an amoeba. It contains Schüffner's dots here throughout the cell. And P. falciparum will overall just look a bit thicker than the ring form, but it will look smaller than P. malariae's basket form. And also deformation of the red blood cell can sometimes be seen. And the P. malariae band form, the way it's spread horizontally like this is a result of the parasite cytoplasm stretching across the red blood cell. So moving on to the gametocytes, P. falciparum has what's called a Leveran's bib, which is just remnants of the host red blood cell that it destroyed. They also have kind of a banana shape to them. P. malariae gametocyte will be round to oval shape. P. ovales will be oval with Schüffner's dots spread throughout the cytoplasm. And P. vivax has very large gametocytes, and they also have Schüffner's dots. So they will be larger than the red blood cell itself. So here is showing the progression of the creation of the gametocytes, known as gametogenesis. So here we just have stage 2, 3, 4, 5, this, which shows the transformation from trophozoite to finalized gametocyte. So it usually takes them 10 to 12 days in the host to mature. And microgametocytes, the male form, can undergo exflagellation within the mosquito due to pH change. So here on the right, you'll see an exflagellated microgametocyte within a mosquito. Now it's possible for exflagellation to occur within a human host sample, but it won't occur within the body. It's usually only going to be if you have a sample that you took from a human and then it's delayed in processing it. So here again is just another more detailed image of gametogenesis, going from trophozoite to the mature gametocyte, male or female form. So the female form of P. falciparum will have more of that banana shape, where the male form doesn't have that shape as much. And here we can just see some anatomical structures of the gametocytes. So stage 1 gametocytes will actually be indistinguishable from trophozoites. And for the stage 5 gametocytes, if you were to stain them using a Gizma stain, the females will tend to be blue and the males will tend to be pink. And these osmophilic bodies are something that only form in the females. So moving on to the ookinete, it is the largest motile cell of the Plasmodium life cycle. It is a result of sporogony, or the maturation within the mosquito. So when the ookinetes are mature, they will burrow into the mosquito midgut to develop into the oocyst. So here we can see the polar ring. In M, we can see the micronemes. And arrows would be microtubules. So again, these organelles are just necessary to help burrow into the host and establish infection. So here's an image of oocysts. The oocysts tend to exist in relatively low numbers, and they're quite rare to actually see them within the midgut of the mosquito. This is probably because the existence of the oocyst is actually dependent on the ookinetes' ability to avoid the mosquito's immunity and ability to survive. For example, in this particular paper where this image was taken, only 4% of the mosquitoes that they tested, they could actually see the oocysts in. So they're just rare to see. So this image just is pretty much a visual summary of the different stages of Plasmodium. You have the sporozoite that the mosquito injects into the human host. You have the oocyst, the ookinete, which invades the mosquito's midgut. You have the zygote, which results from sporogamy, or sexual reproduction between the microgametes and macrogamete. You have a male X-flagellated gamete showing the flagella. There are the gametes themselves, and there is the schizont containing the merozoites. So here, this last table is just a very good summary showing the differences between the different species of Plasmodium and how you can differentiate them. So here, you can see those banana-shaped macrogametes in Plasmodium falciparum. So the ring form of Plasmodium falciparum will have a thin wall to them. There's the trophozoite form, which can deform red blood cells. You don't see it in this image. And the schizont. Here in Plasmodium vivax, the early ring form appears to be smaller. The trophozoite has its amoeboid shape, and the schizont having a medium amount of merozoites inside of it. And here we can see the very large gametocyte of P. vivax that is usually larger than the red blood cell that it infects. Here in P. malariae we see another small ring. Here we can see the band form of the trophozoite. Here the schizont usually has a lower number of merozoites inside of it compared to P. falciparum and P. vivax. And then here we see P. malariae with its very rounded shaped gametocyte. So overall this chart should be very useful in helping you study the differences between different Plasmodium species. So today was a rather short lecture. That's the end of today's lecture, and I hope you learned something, and I hope you have a good day.